First of all, I should say that this has been an extremely enjoyable meeting. I go to a lot of meetings. Some would say far too many meetings, but I always know exactly what people are going to say. But this time was really so different in that I learned a lot, and it's been a very pleasant way of also interacting with students, I think. So I have a plea, because I'm a little bit concerned about time already before I start. So if you have general questions, please could you wait about them till I have interim conclusions. Of course, if it's something you don't understand, you can do it during the talk, but I think it's more efficient if you sort of pack those general questions into one time, basically. So as Roger has already indicated, my background is really in transcription. Actually, my background really in transcription is from Roger's lab, where I finished my postdoc now 20 years ago. It's scary to think about how quickly time flies. So at the time I was in Roger's lab, everybody interested in transcription was interested in only pre-initiation complex assembly and initiation, basically. So pre-initiation really involves the assembly of the general transcription factors. The factors are required for transcription of all RNA polymerase II genes. And the mediator complex, which actually, interestingly enough, is now part of all these general transcription cycle pictures, but was actually discovered in the lab when I was a postdoc there, not by me, but by others. And it was a major discovery that has, of course, changed lots of things, and probably also partly had an influence on Roger eventually getting the Nobel Prize. So we go from pre-initiation to actually the initiation of the transcript, and then we go into the phase that we are most interested in, namely elongation, in which the polymerase, of course, actually makes the transcript. And at the end of the gene, of course, you have termination, and you have recycling of RNA polymerase II, dephosphorylation involved in that, so that polymerase can return to the promoter or another promoter to start again. But as I said, this cycle of pre-initiation, initiation, termination, of those we are really mostly interested in transcript elongation, the actually act of polymerase making the transcript, or as we learned over the last couple of years, traveling the genome, because there's a lot of transcription, a lot of the nascent transcription that really doesn't produce a messenger RNA, but really across the genome there's a constant running of polymerases. And we're interested in this because of the many interfaces this running polymerase has with other processes that occur on DNA. And I'll just point a few of our interest spheres out here. For example, of course, RNA polymerase II has to deal with chromatin. It has to run through these immovable objects. And how does it do that? In itself, RNA polymerase II will therefore also be expected to affect chromatin structure, and indeed it does. So we want to understand and have worked quite a bit on this problem over the years. We also know that RNA polymerase II, of course, will run into transcription obstructing DNA damage. And this leads us to this area that I'll be talking about today, namely this interface between DNA damage repair and transcription. Moreover, there are exons, of course, in genes, and we need to excise the introns and fuse the exons. And this actually happens as the polymerase traverses the gene. It happens co-transcriptionally. So we also want to understand messenger RNA splicing and its coupling to transcription. Finally, these polymerases don't work in isolation in terms of moving machines. Polymerases will clash with polymerases, and we've studied this quite a bit. But moreover, there's also be clashes with the replication machinery. During S phase, transcription doesn't just stop. So we have clashes with RNA polymerase II and DNA polymerase. And it's well known that this is actually very genome destabilizing. This is something we've become very interested in over the last couple of years. And one of the things it definitely results in is recombination, DNA recombination, pieces of DNA that basically travel to places they shouldn't because of these sorts of clashes between transcription and DNA replication. But today, I can't speak about all this, of course. I'll focus entirely on our interest in this connection between transcription and DNA damage repair. So, of course, as you all know, there are many different kinds of DNA damage, and I've just listed a few of the things that will give rise to, for example, double-strand breaks or single-strand breaks or chemical modifications of the bases. And actually, it is amazing how many lesions or damages we have every day. About 2 billion damages per person per day. And it's obvious that we need to get rid of these. Otherwise, we probably would have very fast evolution or no evolution at all. And we have a number of different DNA repair pathways have evolved so that to make sure that only a few of these lesions, if any, lead to mutations or genome instability in more general terms. And as you well know, the repair pathways that lead to nucleotide excision repair, base excision repair, and also mismatch repair was actually awarded the Nobel Prize last year. So our interest is specifically in these bulky helix distorting lesions that, for example, come by sunlight. These are typically thymine dimers or pyrimidine dimers more generally, but for example, cisplatin will also give rise to these bulky helix distorting lesions that are actually interesting to us because they are in a very specific way stopping RNA polymerase II. Polymerase, when it encounters one of these lesions in the transcribed strand, will stop and it cannot go any further. And this, of course, is an unacceptable occurrence. It needs to be dealt with, and indeed the cell has developed a number of different responses to such damage. And I should say these responses are actually specific for this bulky DNA damage. There are other responses to double-strand breaks, for example, which would also be a problem for transcription, but that we can perhaps get back to in the final discussion session. So what do cells do? Well, sort of at the local level, there is transcription-coupled nucleotide excision repair. These damages that are encountered by RNA polymerase II are actually preferentially repaired. Even though they are effectively covered by RNA polymerase II, we know from the structures that the polymerase sits right on top of the damage and the repair machinery actually can't see it. A reaction that we have been very interested in is what we call the last resort pathway, namely ubiquitylation and proteasome-mediated degradation of RNA polymerase II. It's a sort of brutal way of dealing with this problem of stuffed polymerases. And then there's a more global effect, and this is, I think, absolutely fascinating, and not very many people realize this. There's actually a global shutdown of transcription across the cell in response to DNA damage, not just of the gene that is damaged, but globally. There's a trans effect, there's a signal from that damage. Stop transcription, this is too dangerous. And we really do not understand anything about what this process involves in terms of factors and mechanisms. And then finally, this last resort mechanism is that you simply kill yourself. And again, we know very little about the specific role that the elongating polymerase might play in this, but there are some indication that stopped polymerase, if there are too many of them, will actually elicit apoptosis. Again, very little is known about it. I certainly don't know anything about it at all, but it is something we will almost certainly stumble into over the next couple of years. So as I said, really we are in two different modes here. There's a local response mode, this polymerase being dealt with, but there's also a very global or systemic response of the cell to this situation of stress. And really, it can be seen as a simple stress response. And indeed, there seems to be overlaps, for example, with the heat shock response, which it will surprise you to hear, also shows this global shutdown of transcription. You always hear about heat shock giving rise to a lot of transcription, but that is from a few genes. The other genes actually shut down, and we still don't understand the mechanism behind this shutdown. So in my next few slides, I will show you quite a bit of Mickey Mouse science, as I call it, to just get you up to speed with what we've worked on over the last decade or so. Much of that is actually on our work on the ubiquitylation proteasome-mediated RNA polymerase II degradation, and then I'll get much more into these two later. So basically, first and foremost, when RNA polymerase II stops, it elicits this transcription-coupled nucleotide excision repair. Now, nucleotide excision repair is subdivided into two different reactions, what we call global genome nucleotide excision repair, a slow process that will eventually deal with all damage, all these bulky lesions across the genome, and a very specific pathway which only recognizes and deals with damages in active genes, and not only just in active genes, only in the transcribed strand of active genes. And this is because damage in the non-transcribed strand of active, doesn't actually stop polymerase. It ignores them completely. But when they're in the active, in the transcribed strand, RNA polymerase II will stop. And that leads to recruitment of a number of proteins that then leads us into this pathway. So the starting point, the damage recognizing factors are different between general genome repair and TCNER, but the factors that eventually deal with the damage are different. But of course there are special problems involved in having to deal with the polymerase sitting on top of the damage. And this is probably the reason, partly the reason for these differences between this very serious first repair disorder that was discovered, namely xeroderma pigmentosum and Cockayne syndrome. Although we recently shown that actually Cockayne syndrome is a transcription syndrome rather than a repair syndrome like xeroderma pigmentosum. All right, so the alternative to transcription-coupled nucleotide excision repair, or as we call it, the last resort, is actually to simply take RNA polymerase II out and then hope to repair the damage by other means. And the reason for the existence of this last resort is that as RNA polymerase II travels the genome and meets the DNA damage, this really needs to be dealt with. Because in contrast to, for example, replication, where you can dissociate the DNA polymerase and have another translesion DNA polymerase inserted instead and then replicate across the DNA lesion. You can't do that with RNA polymerase II. As soon as you lose RNA polymerase II, you lose the transcript you just made, because the DNA RNA hybrid is actually only 8 nucleotides long. So as soon as you lose this polymerase, you lose the transcript you just made. And this elongation complex in any case is extremely stable. It doesn't just fall off. It can't fall off unless we have a lot of help to do it. So what will really happen here if all these rescue attempts, including DNA repair, fail, is you will have a roadblock and polymerase is piling up one after the other. And this, of course, is an unacceptable situation, certainly if it occurs in an essential gene. So this blocks all transcription of this gene, and we revert to this last resort. And of course, as you can imagine, it is extremely important for cells, if they want to degrade these, to do it specifically. So this is a system that goes with belts and braces to ensure you only degrade the ones that are in trouble. And this is a multi-step process that starts with the recruitment of this ubiquitin ligase and E3 called RSP5 and a ubiquitin conjugating enzyme called UBC5 to the stalled RNA polymerase II. And I should say that this system is completely conserved from yeast to man, and we worked on it first in yeast and now more and more in human cells. And the recruitment of this complex and activated ubiquitin, of course, leads to monoubiquitination of RNA polymerase II. But actually RSP5 is a very, very active ubiquitin ligase, so it will sometimes put a chain on. As it happens, though, the chain that RSP5 puts on is a K63-linked ubiquitin chain, and that's no good for degradation. This chain have a role in transcription, but we don't know what it is yet, if there is any. But RSP5 is associated with a ubiquitin protease called UBP2, and this will immediately proof-read the chain. But we discovered by chemical experimentation that UBP2 is simply unable to take off that last ubiquitin. So basically the end product of the RSP5 UBP2 action is monoubiquitination of RNA polymerase II. So that's the first step of dealing with this stop polymerases. Now we have an exchange or certainly an incoming factor called DEF1, a protein that we discovered many, many years ago and actually got us started on this whole process. DEF1 has a very interesting relationship with RSP5 in that DEF1 actually becomes ubiquitinated by RSP5 itself. And this leads not to degradation of DEF1, it's probably multi-ubiquitination rather than polyubiquitination. And it leads to the proteasome not degrading DEF1, but just clipping it or actually eating its C-terminus. Now this C-terminus contains a domain that makes sure that DEF1 is normally in the cytoplasm. But when it's clipped off, DEF1 accumulates in the nucleus. So now it can really deal with a lot of stopping and stalling that you get in response to DNA damage. So this is a DNA damage response sort of independently of all this that makes it possible for DEF1 to accumulate in the nucleus. But DEF1's role in this reaction is that after monoubiquitination, it actually recruits another ubiquitin ligase. a second ubiquitin ligase that independently reacts in this system. Now we knew, the only thing we knew about this very strange protein DEF1 was that it had a ubiquitin binding domain. And we were fooled for years and years and think, oh, this ubiquitin binding domain of DEF1 must bind this monoubiquitin and thereby be recruited to RNA polymerase II. It turned out that was wrong, and actually, as I said, took us a while to realize that actually the ubiquitin binding domain of DEF1 is instead used to bind the elongin complex, which has a very unusual ubiquitin-like domain. It's not even ubiquitinated, it simply contains a ubiquitin-like domain that makes it possible for DEF1 to recruit it. And once it recruited, the elongin complex now builds on what was already there to make this K48 chain that can lead to the degradation of RNA polymerase II. However, there's yet another fail-safe in this system, in that there is a specific ubiquitin protease again, UBP3, that can remove this entire chain. It can even take off monoubiquitin. So there's a lot of checks and balances in this to make sure that really, do you really want to degrade RNA polymerase II? Yes, we do. And when that happens, when this signal is persistent, we now have recruitment of the CDC48 P97 segregase, which will take RNA polymerase II out of chromatin and feed it to the proteasome. This is not our work, but work by Ray Deshae's lab, who published a very nice paper in 2011 about this. We recently found that actually this P97 segregase requires this cofactor called UBX1, and we are trying to figure out the specific mechanism of this. All right, so this was about the mechanism of ubiquitination and degradation of RNA polymerase II, and actually we worked that out in much more detail than we understand transcription-coupled nucleotide excision repair. There's a lot of sort of unknowns in this process, and it's partly because of a lack of a good reconstituted system. I can promise you we tried for many years, and in the end simply gave up because it was just too difficult to reconstitute this reaction in sort of natural setting that, as Kim was saying yesterday, reconstituted the preferences, the requirements that you know you have in vivo. So in this second part of my talk, I'll be telling you about these two sort of unknowns and our attempts to learn more about them. So basically, as I said, transcription-coupled repair starts with the recruitment of Cockayne syndrome B to RNA polymerase II, and this protein is absolutely required to recruit all these other guys to RNA polymerase II. And actually, in the end, when repair takes place, it's likely that we have this huge complex that contains everybody who you need there. Not just the different repair factors. These guys are the ones that actually do the incisions on each side of the DNA damage. But even the DNA polymerase machinery seems to be there ready to go. We just don't know exactly what happens, because it's obvious that you need to get the RNA polymerase II out of the way, because once you sort of get rid of this DNA damage, you also get rid of the sort of foundations for where RNA polymerase II sit. We don't know where it goes. It's almost certainly, it is not degraded, because the degradation is an alternative rather than a requirement for repair. So we think RNA polymerase II is likely to be moved out of the way, perhaps backtracked out of the way, repair takes place, and then you can continue. But really, this is just speculation. It's been a frustration for many years that we don't really understand. But the key factor for this, though, is to understand the function of Cockayne syndrome B, a switch SNF-like DNA translocase, and of course RNA polymerase II itself in this specific situation. We know more stuff, though. We know, for example, that there are unknowns. That's always nice. We need to find more factors to understand this process. Indeed, just a few years ago, this protein, which is the background for a very severe human disease, which is UV sensitivity syndrome, where patients are extremely UV sensitive. This was discovered quite recently. So we know that there are even more, not just because of that, but for a number of reasons, we know there are factors we just don't have the right, we don't have all the factors yet, basically. We also know there's a very important role for ubiquitination in this process. Indeed, one of our thoughts about the process is that this CSA choline ligase complex is the very large actor to arrive at the scene, and it's sort of a control factor that, okay, everybody's here, we can start repair. So we know that this almost certainly ubiquitinates something in this complex to trigger the reaction. And one of the reasons why we know this is that actually CSB has a functionally crucial ubiquitin binding domain. It must be binding ubiquitin somewhere in this complex. We just don't know where yet. We actually also know that XPG also has a ubiquitin binding domain. Again, we don't know really what it does. We also know that there's an important role for kinase or for phosphorylation. TF2H is a kinase, and among these unknown factors, there is a kinase that plays an important role because we already know from data that I won't get into that phosphorylation plays an important role. So all this just to give you a background for sort of the known unknowns that we have in this system. Now this is at the local level. This is a level of the RNA polymerase II stopping at DNA lesions. At the more global level, we know that there are dramatic transcription changes. This is an old experiment actually by others that shows that in response to DNA damage, you get a actually dramatic decrease, and it's more dramatic with modern tools, I should say, where we almost go to zero within an hour or two in terms of nascent transcription, and then transcription slowly recovers so that we reach more or less normal levels after about 24 hours. In the absence of this protein, cocaine syndrome B, this doesn't happen. And this is not just because CSB is a repair factor. It is also because it is involved in this global signaling pathways to get transcription restarted. Indeed, if we transfect in an undamaged reporter plasmid, this reporter plasmid isn't transcribed after DNA damage. And it stays untranscribed if you don't have CSB, even though there's no damage in this plasmid. So this is really signaling from DNA damage to the whole systemic response system to cease transcription. We also know from anecdotal evidence that there are changes to alternative splicing in response to UV irradiation, probably as a consequence of these changes to transcription. We don't really understand these transcription changes. So the take home from this is there will be both global and local effects of DNA damage, and we really want to understand this whole system, and we want to understand the factors and the mechanism that underlie these fascinating consequences of DNA damage. Now, the classical way of doing this, the way I was taught when I was a postdoc, is to reconstitute, then purify, purify, purify. And as I said, when I started my lab, this is what we did, and we failed miserably. We couldn't reconstitute even something as simple as transcription-coupled nucleotide system repair. It made us realize we have to do it in another way. And what I'll describe now is sort of where we've evolved with this, because we try to do this by interactomes and omes in general, genome screening, both at the protein and gene level. So basically, the stories I'm going to tell you today is about this screening methods to find proteins involved in this, what we call the transcription-related DNA damage response, really just ranked on what we think are their probability of involvement. We also call it Googling the proteome because, as you will see, we actually interrogate this process from a number of different angles and just take the best responders. And this has led us to a fascinating gene and protein complex encoded by the ASCC3 gene, which I would claim is a completely new type of gene, or we call a MANC gene, because it's not just a messenger RNA. So how do we do this? So basically, we start out, rather than purifying through homogeneity and knowing exactly what we've got in our tube, we look at interactomes. And I told you that the key factors to look at is RNA polymerase II itself as it sits at DNA damage, and then this factor called cocaine syndrome B or CSB. And then we use SILAC proteomics, in which we use different heavy or light lysine and arginine in our reactions, and we have untreated and UV treated cells that we can compare in the same mass spectrometry reaction. So we basically pull down RNA polymerase II before or after UV, and we find out what interacts with it specifically in response to DNA damage, and the same for CSB. Likewise, we map post-translational modifications. In this case, specifically ubiquitination and phosphorylation that occurs as a response to DNA damage. Sometimes you have much more ubiquitination, but actually sometimes you have much less. Then we also look at chromatin proteome, and the reason why we do this is again informed by what we already know. We know that CSA, this control factor, only gets stably associated with chromatin in response to DNA damage. Otherwise, it's found in the nucleoplasm. So we actually also look at the chromatin proteome, and I should say that these interactomes and so on are actually starting from chromatin. So we don't just start from the general cell extract, we start specifically from salt-soluble chromatin extracts. So this also acts as our sort of our input for this. Now these are all of course protein based assays. These are all select proteomic approaches, but we perform them under identical conditions, 3 hours after UV, in the hope of not just catching the immediate response to UV, but also catching sort of this recovery after DNA damage, after repair, when transcription has to get started again. So this gives us a much broader range of proteins, both immediate response genes and late response genes, or proteins, whatever you want to call it. Importantly, we also use this proteasome inhibitor, MG132, to further investigate the importance of ubiquitination and proteasomal degradation for this. Now this is combined with an siRNA screen in which we look for genes that affect transcription after DNA damage. And I'll get back to this in much more detail very shortly. And then the idea is basically a Venn diagram idea is, oh, if you score here and here and here and here, you're likely to be important in this response. So basically, that's what data integration means. And we've actually developed what I think is a marvelous interface where you basically can plot in whatever gene you're interested in and investigate how it's scored in our screens. I think this is the way that all these sort of screens should be presented, but arguably because we have so many screens, it was also extra important in this case here. And although I don't really like network analysis at all, it's against my training, we do some network analysis to see which protein sort of groupings light up in this sort of analysis. But we're much more interested in the identification of individual hits, because we really want to get on to understand the mechanism, the role of these individual new factors involved in this process that we could have found in the old days by reconstituting and purifying to homogeneity, but which we now get by sort of interactomes and actually functional importance in this system. So this is, I'll just very quickly show an example of what these proteomic interactomes look like. This is the CSB interactome in response to UV radiation. You can see that I use both in the absence in the presence of MG132, in the absence of MG132. So the ones up here are the ones that interact much more with CSB after UV, whether you have proteasome inhibition or not. So this is sort of a very well-controlled, very select crowd of proteins. These are the guys that really do interact with CSB only after UV, or mostly after UV. So if we focus in on that, we can start looking at what factors are there. And I should say that these green and green and red ones are the ones that also scored functionally. So we know that not only do they interact with CSB, they are also functionally important, because when we knock them down, that does really matter. So we're really pleased to see that the most dramatic hit we get here was UVSSA. And at the point when we found this, UVSSA had actually not been published yet. So we knew immediately we were onto a good screen here. We also found the CSA-containing choline ligase complex. That's sort of expected and known that CSA and CSB functionally interact. So again, this is a very nice control for this actually working out. We also found many subunits of TF2H. The factor that I actually worked with when I was at Roger, it's sort of known that TF2H has a very important role in transcription-coupled nucleoside repair. It makes sense that it interacts with CSB. But then, of course, we also found completely unexpected stuff, and that's what we're really going for. We don't really care about this stuff that we know about already. But we found the integrated complex, which is a complex that binds the CTD of RNA polymerase II and is known to be involved in termination of transcription. Now, we don't understand this at all, but of course now we're working on the integrated complex in the context of this process, in the context of the DNA damage response. So now that's just an example of the proteomic approach. So the genomic approach is, of course, a completely different one. Here, we take the 20,000 genes across the human genome, and we try to knock it down individually and see which ones matter. So this is just our assay, really, which is an assay that is microscopic in the sense that we simply look at Diaby staining and then nascent transcription, which we measure by ethyl uridine incorporation 20 hours after UV. So this is quite late in the process. This is how it looks in wild-type cells. You will see some cells that transcribe more than others. You will have a normal distribution, as you would expect for any biological system. But siRNAs that lead to what we call low transcription phenotype look completely different. You can see here very little nascent transcription 20 hours after UV radiation compared to the normal control condition. Of course, we don't know what the cause is. It could be that there's absolutely no repair, and then of course you don't recover transcription. But it could be that you have normal repair, but you knock down a factor that is required for this transcription recovery to occur. Or it could of course be that you have lower overall transcription because this screen, because that's the only thing that's possible, is only done plus UV. We don't do the control minus UV. It simply isn't possible. But at this point, we don't really care what the cause is. We just care about getting some hits that we can investigate further later. Likewise, we got these fascinating high transcribers, knockdowns that lead to much higher transcription than the wild type. So it's as if transcription has become, it doesn't care anymore about DNA damage being there. You just continue to transcribe. Of course, again, there can be a number of different possible causes, and again, we don't really care about what those causes are. We just care about getting hits to investigate in our screen. So basically, as I said, we distribute these into high transcribers and low transcribers. It might seem like there's a lot of these and a lot of those compared to the total, but this is sort of a hill going to many thousands of genes. Here actually you have 18,000 in total, and we have postulated 539 high transcribers and about 1,000 low transcribers. As you'd expect, proteins like CSB score as low transcribers. If you don't have CSB, you never recover transcription after UV radiation. So this is an important positive control. We don't actually have a positive control for these guys up here, and we don't really know what to make of them, but we're certainly working on it. So the idea with this way of screening is really that rather than doing one screen extremely well, like in the old days, purifying to homogeneity, you can do that in your screening, or let's say in a single screen as well, but you can't really do that when you do this many screens. So we only do them very quickly and dirtily if you want, because this is very time consuming and also actually money consuming if you know what I mean. But the idea here is to focus on the stuff that scores again and again. This, of course, we give if a protein both interact with CSB, becomes ubiquitinated after UV, becomes phosphorylated after UV, transfers to chromatin, scores in the siRNA scheme, or interacts with RNA polymerase II, you pretty much know this factor is worth looking at. Of course, it's very unlikely. There's no reason why you need to do all these things at the same time. And we'd argue that even if we take away some of these, we still have a very high likelihood of actually being worth pursuing if you just score on a couple of these screens. And indeed, we've had the attitude that anything that scores proteomically convincingly and has an effect when knocked down is certainly worth pursuing. And this has actually turned out to be extremely helpful. Indeed, my whole lab is now working on hits from this screen, and we have high confidence in these hits because we already know something about what they do from the screens themselves. Now, one of the big challenges was actually how to score this. In the beginning, we just did this very primitively. We had these, let's say, these 6 screens, 6 different ways of scoring. For example, if you interact with CSB, you get 1 point. If it changes upon UV, you get another point. If you interact with RNA polymerase II, 1 point. If it changes after UV, another point, and so on and so on. You could, in this screen, get a total of 8 points. And we get this sort of distribution where, of course, a lot of genes will score in one of these many screens. And you can't investigate 10% of the genome. It's not really a screen anymore. It's just a huge ensemble of proteins. But as you can see, we really wither down these to very lower levels when you start to require that they score in several screens. Now this, of course, is a binary score. How do we decide whether something actually interacts with CSB or not? Now this is all based on set values in terms of how much you interact with CSB, how confident we are about you interact with CSB. So this is another way of scoring these hits rather than a binary system. So the alternative here is to simply aggregate all these set scores from the individual screens, add them up, and then actually we used computer-optimized weight factors based on the known transcription coupling nucleotide excision repair factors. So we trained the system to recognize how to best hit these guys. And it turns out that, as you would expect, the genetic screen found these very efficiently. So we gave that screen very high weight in scoring this. Likewise, somewhat to our excitement, ubiquitylation turned out to be a really good predictor for involvement of these guys in the system, and likewise interacting with CSB actually. So this basically tells you that a very, very few proteins have a very high score. That's that large peak here. And the majority, of course, score here. You can see this accumulation of these known TCR factors up here at the top. Some of them actually score fairly lowly, but that's sort of beyond the point. That's the sort of the downsides of this sort of scoring system. So what are some of these hits? I don't have time to go through actually very many of them at all. You see again the integrated supercomplex that I mentioned interacts with CSB. It scores again and again. SCAF4 is a termination factor again that we are working on right now, and a number of other proteins I don't need to get through, but we are pursuing many of these now. And later on, I will, as you heard, be talking about the absolute top scorer, ASCC3. But we are also working on this protein called STK19 because it was one of those completely unknown genes. Nobody's ever studied it to any significant extent, even though it has this very interesting name. I should say this name might be a misnomer. It might not be a kinase at all, but that's beyond the point. The reason why we got excited by it is it actually, beyond scoring very highly in our screen, it's a cancer gene. It's a cancer driver from not only from the broad, but also from a study in Australia. So this is just the take-home message from this paper in Cell that found STK19. It's clearly a melanoma driver, and of course melanoma is really nice in terms of UV damage because that's exactly the sort of cancer we'd be interested in, but really nothing was known about what STK19 does. And now we place it in the DNA damage response. It is important for recovering transcription after DNA damage. It interacts with CSB after UV radiation, and we know quite a bit about it. We don't need to do, we didn't need to repeat all those experiments to learn what STK19 did. So we simply looked at, so does it actually participate, likely to participate directly in the reaction, or is it it might even be a plasma membrane protein that very indirectly participates here. But what we did in collaboration with the Conway lab was to basically express GFP-tagged STK19 is actually vastly overexpressed in this case, but this just gives you an idea of how it looks. It's really found in the nucleus of cells, and I'm just showing you areas here that Joan and her colleagues laser irradiated to mimic UV damage, and then very quickly after DNA damage you can see that STK19 accumulates in these specific areas. So it is recruited directed to DNA damage. But that's about as much as we know about this protein now, and as you can imagine, there's a postdoc working very hard to figure out the specific mechanism of what STK19 does. It's a fascinating protein. It doesn't go to DNA damage immediately. Actually, it's very, very unstable, almost undetectable under normal conditions. Then it becomes stabilized by DNA damage and recruits to DNA damage after about 8 to 10 hours. We have no idea what it does, but we are fascinated by its completely new role in this response, and of course by the fact that it's actually a cancer driver. It's important for human disease. So this brings me to my first interim conclusions. I told you that you can use this multi-omic approach, or Googling the proteome, as a very powerful tool to discover new factors. And this is for any biological process. This is not restricted to the process we're studying here. Indeed, we would challenge anyone to try and find a better way of doing this than this multi-omic approach. And we discovered in this process this new very poorly studied melanoma driver, STK19, that we basically know very little about at this point. Okay, so besides having investigated specifically this screen for new factors, we also wanted to understand the mechanism, what actually happens at the genome-wide level to transcription and messenger RNA splicing. So this was a collaboration between several people in the lab looking at nascent transcription before and after UV radiation, and we use a technique that I'll get into in a bit, and also then just look at splicing, what happens at the genome-wide level to splicing in response to DNA damage. So a number of years ago, we developed a technique that we call Diaby grow-seq, and you remember from my very first slide the transcription cycle. What Diaby does is basically to stop the transcription cycle at the initiation level, all the polymerases will have initiated, but Diaby, when you have it present, will not allow you to transfer to the elongation stage. So the polymerases that are already elongating will run off the gene, but Diaby will basically accumulate all the polymerases close to the promoter in all genes. Now, when we now remove Diaby, the polymerase will run in a synchronized manner across the genome, across genes. And you can see this very beautifully here. So this is 10 minutes, 25 minutes, and 40 minutes after release from Diaby inhibition. And so basically we wait for 10 minutes and then we give a 5-minute pulse with BIU, and then we sequence what we have, what transcription we have after this incubation. And you can see that after 10 minutes, of course, you only have transcription very close to the promoter. I should say that this gene is transcribed in this direction, of course. So you only have transcription very close to the promoter. And then after 25 minutes, of course, you can see that it's sort of what we call the wavefront of RNA polymerase II. It reaches much further into the gene, but of course you still have new initiation. These are the guys that we have down here. So again, we wait 25 minutes and we look for 5 minutes what transcription is going on at this point. And again, after 40 minutes. Now, if we UV irradiate just before releasing from Diaby, what happens then? Very, very different result. The immediate response is nothing. Really, you still have good transcription at the beginning of the gene and the beginning of the response. But very quickly you see that whereas we reach very fine to the gene after 24 hours in the absence of UV, really polymerases sort of slow down. And there's still transcription in here. Remember again the way we do these, we wait for 25 minutes, then we measure transcription for 5. So there's lots of transcription close to the promoter. Actually that doesn't really seem to be affected. This is not just a slowdown of elongation, this is also gene spatial restriction of transcription to the first about 25 kilobases of the gene. If we look at this at a metagene level, so average and across all genes we can measure in the genome, you see something like this. You see a very strong peak of transcription very early in the gene. You can see how polymerase is spread further and further into the gene with time. And we can call the wavefront bioinformatically to 10 to 12 kilobases into the gene after 10 minutes, 45 kilobases after 25 minutes, and about 78 kilobases on average after 40 minutes of transcription. In contrast, after UV, you can see the distribution is completely different. The wavefront is the same after the first time point, and very different already after the next one. And then you really don't move much further. It is as if you sort of hit a wall and you're not allowed to transcribe much further, even though there's plenty of activity in the beginning of the gene still. So transcription continues to be active near the promoter, but you don't get very far. So there's both a very strong slowdown of elongation, actually to less than 5% of normal speed. We can calculate that also because we've extended this to 24 hours. We can see that elongation really is the last thing to recover to normal levels. But at later stages, initiation is very dramatically affected as well. But the immediate response to DNA damage is a slowdown of elongation and a restriction of transcription activity to the promoter proximal area. Now, we combine this with looking at alternative messenger RNA splicing. So basically this involves deep sequencing of total messenger RNA before 8 and 24 hours after UV radiation. And then we ask, so what are the events that we see most frequently? And of course you get a long list of different events, and I'm not a splicing person, so many of these I actually look into what they are. That's not so important. We knew already from the beautiful work of Arthur Kornblitz's lab in Argentina that one of the things that definitely happen is you have quite a few exon inclusion events. These are exons that are typically not included in transcripts, but that after DNA damage start to be included. And it's always been argued, oh, that's probably because elongation is slow after DNA damage. And since splicing happens co-transcriptionally, the slow elongation gives the splicing machinery time to recognize these weak intron-exon junctions and include exons that are normally not included. Now, however, we saw just as many new exon exclusion events as we saw inclusion events. So this sort of kinetic model is probably not quite as straightforward as it seems. However, the most frequent event we saw by far was alternative last exons. Now, as I said, I'm not a splicing person. I had no idea what alternative last exon splicing was, so I presume that you guys probably don't know quite either. So let me explain it to you, because it's fascinating. Lots of genes have alternative last exons with their own polyase signal, their own termination sequence. And it actually switches between the two forms. So in this form, for example, you simply skip this exon. Very often the reason why you don't read this polyase signal is because transcription is, splicing is co-transcriptional. So you actually, the transcript is spliced as you go across this gene, and the polymerase apparently doesn't recognize the polyase signal and simply runs to the end and makes a much longer transcript. So in this case, the blue last exon will be excluded. Now, the other alternative is that you do recognize this polyase signal and you terminate here, so you don't actually read the rest of the gene. And what we saw in response to UV radiation, as you can imagine, was we saw this much, much more frequently. The vast majority of UV upregulated alternative last exon isoforms reproduce from much shorter pre-messenger RNAs. Now, I'll remind you that, of course, we also have this restriction of transcription to the beginning of genes. So you could say, oh, it's no surprise because you can't actually make the rest, and that is exactly what happens. So this is just one example of our sequencing result. Of course, this is deep sequencing across the messenger RNA we get, and these stipple lines are simply the non-existent introns because we don't read those. This is messenger RNA. So this is the read density across the HERG4 gene. And you can see that in the annotation of the HERG4 gene, there's both a very long isoform and then a much, much shorter one. And after UV, you can see a very dramatic upregulation of this shorter one, which has a unique terminal exon that just doesn't exist in the long isoform. So it's very easy to detect it. Now, this normalizes after about 24 hours, so this is a transient upregulation, and at the same time, I should say, very dramatic downregulation of the rest of the gene. So actually, the relative amounts of these two transcripts is dramatically different. You can also see these when we use these exon-exon junction qPCR reactions. If we set them both to 1, the long and the short isoform before UV, you can see that 8 hours after UV, the long isoform is dramatically downregulated and the short isoform is dramatically upregulated. So the difference between them is really very, very dramatic. And this sort of normalizes after 24 hours. Now, the production of these alternative last exon isoforms happens during this period when transcription is gene spatially restricted and very slow. So it's almost certainly because you simply don't make the rest of the gene. That's why you switch to these short isoforms that have an alternative last exon, basically. So as my second interim conclusion here, I told you that the global transcription shutdown is primarily, or at least initially, an elongation shutdown. Polymerases start going very slowly, and you sort of restrict transcription to the beginning of genes. Now, this actually explains an age-old conundrum. That is that some genes are actually highly upregulated in response to DNA damage. But these genes are all very short. And this now makes sense. You cannot upregulate a very long gene because you never get to the end of it. All these genes, FOS and JUN and all these sort of stress genes, they are all very short genes, almost without introns. They've evolved this way. They need to be short because otherwise they would never be upregulated. This progressive reduction of elongation speed suggests that it's an actively induced process. It must involve signaling, probably to chromatin or to elongation factors. And indeed, you remember these high transcribers from our screen. These are probably knockdowns that sort of bypass this whole shutdown. They just continue as if nothing happened. And we are therefore very interested in these high transcribers because they probably are bypassers. And as I told you, RNA polymerase II transcription is also spatially restricted. I think this is really important. Only the promoter proximal 20 to 25 kilobase are efficiently transcribed. And now this, of course, we think there's a direct correlation here, a direct consequence, that this decrease in elongation speed and scarcity of nascent transcripts likely results in these alternative last exon splicing events, creating new, shorter messenger RNAs. Okay, so the next thing is, does this matter? Do we care? This might just be something freak that cells do and we don't care about it at all. So it is important to establish whether this is of physiological importance. But I'd like to take questions now if there are any.
Yes, I'd like to ask, so when you irradiate with UV, do you have an order of magnitude for how much damage you have?
Yes. very good question. So there's a damage on average every 10 kb. Okay, so it's not like you have lots of DNA damage. I should also say, over the 24 hours after UV radiation, we have very little cell death. So it's not like, oh, all these cells die, so this is a crazy. So this is moderate levels of DNA damage. Yeah.
So is the polymerase transcription limited because of lesions or because of regulation?
Both. Really, really important. Both. Of course, polymerase stop DNA damages, and that will lead to some of this. But the fact that we can actually bypass this, and I didn't really say that, we know some of our high transcribers ignore damage and transcribe much further into genes. So it's not just stopping a damage, it's also this trans-signaling, regulation, as you say. And we think actually it involves ubiquitylation directly of RNA polymerase II, and we even have ubiquitylation sites. So this is something we're working on, but that's sort of the downstream mechanistic interrogation of this system. We need to know the system first. We need to know the questions we need to ask, and we know them now from this sort of genome-wide phenotypic analysis, if you want, molecular phenotypic analysis. We know what questions to ask. But you're right in asking, is it just damage? No, it's not just damage, it's more than that. Yeah.
Does the global level of PO2 go down?
No, not well. In response to DNA damage, as I told you in the beginning of my talk, you have ubiquitylation, degradation of RNA polymerase II, but only actually the polymerase that meets DNA damage. You don't touch the polymerase in the nucleoplasm or even the promoters. The way this is regulated, at least partly, is via CCD phosphorylation. So we've shown that the elongating forms of RNA polymerase II, the ones that meets DNA damage, are prone to be degraded, to be recognized by RSP5, whereas the initiating forms are not. Okay, so to put a very simple spin of that.
And we recover the global transcriptions as splicing profile.
Yeah, so I actually showed that in the data, and I'll show you again, that splicing normalizes after 24 or sometimes only after 48 hours. Yeah, so this is a transient response, also the splicing level. And the question really from that is also, does it matter? And I will show in the rest of my talk it's very important. And the way I'll show you this is by focusing on what actually is our top hit from the multi-omic screen, this protein called ASCC3. This is a protein that I'd never heard about before, but I certainly should have, because it's, in many ways it's like mediator. It's a co-repressor and a co-activator for transcription across the genome. It's just been sort of overlooked. It was discovered 10 years ago by a Korean group, and really there are 3 or 4 papers on this protein and this complex that it's in. So why did we discover ASCC3 in the multi-omic screen? It interacts with CSB, it interacts with RNA polymerase II, it is highly ubiquitinated in response to UV, it is highly phosphorylated in response to UV, and its knockdown affects transcription after UV radiation. And as I said, this is a very poorly studied protein and complex. It's part of this activating signal co-integrated complex, or ASC1 complex. It's subunit 3, so we also have ASCC1 and ASCC2. It's both a co-activator and a negative regulator of transcription, and this is sort of really, it's very poorly understood, and of course it's one of the things we really need to understand now, is what is the role of ASCC3 and its complex members in transcription. But the reason why we got interested in ASCC3, beyond the fact that it's the top scorer, is the way it's scored in our siRNA screen. Because we used this commercial Dharmacon pool, and it turned out there were 2 different pools hitting ASCC3. Now this is somewhat unusual, because you have 20,000 genes, that's enough to deal with, to knock down, and Dharmacon has 4 siRNAs in each pool that will knock down each of the 20,000, but there were 2 pools, 2 separate pools for ASCC3, and they scored differently. One scored ASCC3 as a high transcriber. When you knock down ASCC3, you get higher transcription after UV radiation. But the other pool scored ASCC3 as a very low transcriber. When you knock down ASCC3, you have very low transcription after UV. Now this is either weird or wonderful. And of course I wouldn't be here telling you about it if it wasn't because we think it's the latter. Because it actually turned out to be because of alternative splicing. So ASCC3, you can see the annotation here, has a short isoform which is dramatically upregulated relative to the long isoform after UV radiation. This is a very, very long gene, 375 kilobases. It encodes a very long helicase of unknown function that is a 200 kilodalton protein all in all. The short isoform, as you can see, is only about 20 kilobases, so it still fits this, restricted, gene spatially restricted area. And indeed, what we see after UV radiation, and this actually only happens quite late in this time course, is that we have a relative upregulation of the beginning of the gene compared to the rest of the gene. You can actually see that the rest of the gene is barely transcribed at all, okay, relative to the beginning. And you can see this most clearly when we do these comparisons where you set them both to 1 and times 0. And you can see that after 24 hours, there's really a very, very big shift to the expression of this short isoform. But it's not so that the short isoform is not normally expressed. It is normally expressed. It is just that after UV radiation, there's a shift in the balance between them, so you almost only express the short isoform. So what are these isoforms? So this is the long isoform. The blue here is, of course, the open reading frame, the coding sequence. And you can see here the white siRNAs from the pool, and you can see how it scores. When you knock down with these, you get high transcription after UV radiation. In contrast, this second pool had a couple of siRNAs that are actually specific for this short isoform of ASCC3. And these are the ones that give rise to this low transcription phenotype. Actually, when we deconvolute this, we can see that specifically these short isoform specific ones that give rise to this phenotype. We were really lucky with this because Dharmacon in the later iterations of this commercial pool have left out this second pool. We would not have discovered if we did that experiment today. We were really lucky to find it. But it really has this dramatic difference between the short isoform, which as you can see encodes a very small protein, and the long isoform, which encodes a very big one. Now we also investigate the, what's going on here? Oh yes, we investigated what is the consequence of knocking out the partners of ASCC3. So now we're looking at very specifically the protein complex containing this helicase. And as you can see, knocking down ASCC2 and ASCC1 also gives us high transcription after UV radiation. So this is sort of a repressor complex. It keeps transcription repressed after UV radiation. When you lose that complex, you overtranscribe as a consequence of UV and the recovery of transcription. So this is just to give you a sort of example of how we do these sort of measurements. We measure across this microscopic slip of cells. This is after no UV. You can see we get this normal distribution of cells that transcribe more or less as you'd expect for any cell really. And as you can see, already 1 hour after UV radiation, we almost completely lose transcription. Whereas there's only 4.2% of cells in this, what we define as a lowly transcribing fraction. That's almost all cells after 1 hour of UV radiation. This is very dramatic. It's almost all due to the stop of elongation. You really transcribe very, very slowly, and it gives rise to this transcription shutdown, as we call it. And then already after 6 hours, you can see some transcription recovery, and you see this bimodal sort of behavior of cells, cell population, after 24 hours of transcription recovery. Now what happens to these profiles, of course I just showed you this, is that 20 hours after UV, you see this considerable proportion of cells remaining in the lowly transcribing fraction, even the control siRNAs. But you see this immediate dramatic transcription response as well. Now when we knock down the short isoform of ASCC3, the one that gives rise to a very, very small protein indeed, if at all, you see that we simply remain in this lowly transcribing fraction. Almost none of the cells recover normal transcription. And this is exactly the profile we get when we knock down Cockayne syndrome B, sort of the classical transcription restart protein. So this really, these cells without this short isoform cannot restart transcription after UV radiation. They shut it down normally, there's no problem with that, but they cannot restart it after UV radiation. So this short isoform is required for transcriptional restart. In contrast, when you knock down the long isoform, we do get the normal immediate response to DNA damage. Transcription is shut down, but this protein this complex must be required to keep transcription repressed, because you can see that when we lose it, first of all, we lose even this fraction of lowly transcribing cells, and most cells move to a much higher transcribing fraction. So this really is a general repressor of transcription in the late phases of the UV damage response. I should just point out again, the immediate response, the immediate repression transcription, ASCC3 complex is not involved in that. It's only involved in sort of maintaining that level later on in transcription. So ASCC3 long isoform is not required for establishing repression, but helps maintain it after UV radiation. So these two splice forms from the same gene have opposite effects on the DNA damage response. It's really quite remarkable, at least we thought so. So we also looked at this. Now, what I've shown you so far has been nascent transcription. We just measured transcription. We don't know what gene we're looking at. We don't even know whether this leads to a messenger RNA outcome, because, of course, it's very far from running across a gene to actually making a stable messenger RNA. So we thought, well, we'll also look at stable messenger RNAs 24 hours after UV radiation. We can do this by simply putting them on a gene expression array like old-fashioned technique, so to say. And when we knock down the short isoform, we see a number of genes, hundreds of genes, that are dramatically downregulated compared to wild type. That's why they are blue. Now, when we interrogate these exact same genes after knockdown of the long isoform, you see that the same genes are highly expressed compared to wild type in this knockdown. So this sort of mimics what we saw at the global nascent transcription level. At the level of these individual genes, we see the opposite effect of these two knockdowns. They behave oppositely. And I'll just show you a couple of examples of that. See here that when we knock down the short isoform, this actually is a gene, as it happens, this is a gene that is highly expressed or induced by UV radiation. This is not true for all genes, of course. Actually, the majority of the ones I've shown you so far were not that case. But this is easier to appreciate here, where this is a highly induced gene by UV in wild type, and it isn't that highly induced in the short isoform knockdown. Actually, very little induction. You can see that more dramatically here, for example, where we see this induction of this gene, but it doesn't happen in the short knockdown. Actually, this is a knockout cell where we specifically with CRISPR knock down that first alternative exon. And we see the opposite effect when we use shRNA to knock down the long isoform. I should say the long isoform is essential. You cannot knock out this gene. It's an essential gene. So we have to just go with these partial knockdowns you get with shRNA. But you can see that in contrast to the effect we get up here, where the black axis is now, the black bar is now the same as here. The y-axis is different. You can see we have over-induction of these genes in the knockdown of the long isoform. So they have opposite effects. So one possibility is that this is a little module that self-regulates. So we asked the simple question, if we first knock out the short isoform, can we rescue the terrible phenotype of that by knocking out the long isoform? So can we remove this completely from the picture? And that actually turns out to be so. So this is just one example of that. You see this DDX60 gene, which we set to 1 in terms of its behavior after UV radiation. This, as it happens, is an induced gene. It becomes upregulated in the wild type cells. We set that to 1. But in the knockout, short isoform knockout, you can see that this gene is no longer induced. However, if we, on top of this mutation in this knockout cell, remove the long isoform, it starts to behave much more like the wild type. Okay, so the lack of the short isoform is only a problem when the long isoform is present. The same is true actually when we look at a global level. Here the fraction of lowly transcribing cells goes dramatically up when we knock down or knock out the short isoform. This is what we have shown you already. The fraction of lowly transcribing cells shoots into the air just like CSB knockouts. And we can sort of rectify that by knocking out the long isoform. And this is true both when we do the knockdown of the short or the knockout of the short. So basically, in the absence of the long ASCC3 isoform, or I should say messenger RNAs encoding other members of the ASCC complex, this transcription suppressing effect of the short isoform is negated. So these two isoforms regulate each other. So this now brings me to the protein level, which of course in the context of this conference is extremely important, and I'm sorry about just talking about DNA and RNA, but here we'll get to a little bit of it at least. So this is, as I said, a helicase which functions in the context of the ASCC1, ASCC2, ASCC3 complex, and this doesn't really have anything of any interest. It's just a weird short piece of DNA. And of course, we tried to rescue the phenotype of knocking out or knocking down that short isoform by simply, as you normally do, making a transgene, the coding region and a promoter, re-express and see what happens. So basically we can do that. We can put the cDNA into the cells. We produce very nicely the expected 111 amino acid protein. Does it rescue the phenotype? No, it doesn't do anything. We did this again and again and again because it had to work. You must have done some mistake. It can't be right, but we really clearly expressing this protein. Now, in the end, we decided, well, perhaps we should just give it this as well. It might be something about the 3' untranslated region. This still gives rise to 111 amino acid protein, of course. That doesn't change. And importantly, this is really important, the levels of this 111 amino acid protein was not different between these two cases. But now it rescues. So this indicated that it's not the protein. The protein doesn't matter. It's this RNA, this non-coding RNA that matters. And we can test this. We can simply insert at the beginning of this open reading frame a stop codon. So now we no longer get any protein because there's no open reading frame. It is stopped before it even gets started. And we indeed get no protein, but we still get rescue. So this is rescue based on the RNA. Indeed, this is a non-coding RNA. And even though it's the short isoform, it's a long non-coding RNA, a link RNA, because it's actually almost 3,000 bases long. Okay? So this is all, in all likelihood, a structured RNA that becomes expressed as a result of alternative splicing and has a role in this DNA damage response separate from protein. Indeed, when we look at where this RNA is, it's in the nucleus. As you can see here, the short, long non-coding RNA is, when you overlay it with Diaby staining, is in the nucleus. And actually, we find it mostly in these spots, and we don't know what they are yet, but we actually, in other better pictures, see it generally in the nucleus. There's a little bit of it in the cytoplasm, but really the vast majority is nuclear. In contrast, as you'd expect, of this long isoform, which is non-coding. Of course, you find some of it in the nucleus because it's actually being made, and it takes a while to make a 400 kilobase long piece of RNA. But the vast majority, as you would expect, is in the cytoplasm because it's involved in translation. I won't have time to go into this, but actually we even investigated where in the nucleus is it. As you'd expect from a long non-coding RNA, the stable isoform is chromatin associated. Okay, so this is a RNA that regulates transcription, and it is transcribed from the same gene as an important protein that has the opposite function. Importantly, this knockout or this knockdown of this short isoform gives rise to UV sensitivity. So it is very, very important because we have lots of things that are involved in the DNA damage responses, and they don't always give a lot of response to the damage you actually impose on them because they are backup mechanisms. But in this case, as you can see, these cells are really UV sensitive. It is physiologically really, really important to have this short isoform.
So is the localization affected by UV radiation?
Not really. Not really. But of course, you accumulate much more of it after UV radiation, but it's not about localization. Okay, so really this is a talk without much mechanism, and we will over the next couple of years, I promise you, be working on that precise mechanism. But what I told you really is that UV radiation results in numerous alternative last exon events that create new, much shorter, I call them messenger RNAs, but as you just saw, actually it's not in this case of ASH3 a messenger RNA. And one of these occur at ASH3, and the different splice form have opposite effects. The long isoform is part of the ASH3 complex and is important for maintaining low transcription long after UV. It's a transcription repressor that maintains transcription low after DNA damage. The short isoform is completely different. It's an RNA isoform. It is technically a messenger RNA because it does have an open reading frame that can, in theory, express a protein, but it contains a 3' UTR and non-coding region, of course, which is important for the UV start of transcription after DNA damage. And this ASC-C3 protein and the RNA cross-talk, and in other words, they regulate each other's function. And this actually is the first example of a gene expressing both a protein-coding messenger RNA and a non-coding RNA from the same promoter in the same direction, and of course with opposite biological function. So we propose to at least call this gene, or we think or hope or dream, is a founder gene of this group of genes called messenger with an alternative last exon non-coding RNA or MANC RNA genes. And we've actually found more than 70 other genes like this. I didn't really focus too much on that, but I told you that alternative last exons are a very frequent event. And these 70 genes are actually scoring very highly in our multi-omic approach. So they are really connected to the DNA damage response. We don't know what they do there. We do not know whether these short isoforms matter or not. But as you can imagine, we are working on that as we speak. And then finally, just to give you an overview of what I hope I've shown you today. Under normal conditions, we have, of course, fast elongation. This leads to a sort of skipping of this alternative last exon close to the promoter, and we predominantly express this ASCC3 long messenger RNA that leads to ASCC3 protein incorporation to ASCC complex. After UV irradiation, we have this dramatic deceleration of transcript elongation and sort of spatial restriction of transcription to close to the promoter. And long after UV radiation, you really need this complex to maintain the transcription shutdown. Without this complex, without ASCC3, you don't maintain low transcription and things just go awry. We actually don't know what that means. We think it's to maintain transcription in a low state to avoid mutations, but that needs to be proven. Because you'd wonder, why do you need to maintain transcription low even 20 to 25 hours after UV radiation actually occurred? So this spatial restriction of transcription is certainly important in terms of inducing these alternative last exon splicing events that now shifts the ASCC3 gene so that now predominantly expresses this short non-coding RNA, which is a stable RNA. And this, in the context of this whole system, is incredibly important for transcription to recover after UV radiation. Without this stable RNA, you really stay at this state for much, much longer than you should. But once you have it, you can recover normal fast transcription. I've also told you, very importantly, that this short stable RNA almost certainly regulates this ASCC3 complex fairly directly. We've been trying desperately to show that this stable RNA binds to this complex, but we've so far been unable to. But that might just be a technical problem, because it's actually not easy to work with stable RNAs and look for protein-protein interactions. But there are other possibilities. Stable RNAs can have an effect of chromatin structure and a number of other events that are important to recover transcription. So really, in terms of the precise mechanism for how this works, we don't really know yet. And just finally, the people who actually did this work, this was a three-way collaboration with Stephen Boeing being responsible for all the proteomics and the website that I talked to you about in the multi-omic approach. And Laura Williamson, an unbelievably talented postdoc, did all the work on the siRNA screen and all the work on ASCC3, and they had help from Marco Seponaro. And of course, I should also thank all the absolutely fantastic facilities we have at the Crick Institute. This is really a scientist's wet dream, because we have all this help with mass spec, with high throughput screening. They don't do it for us, but the postdocs and students can go to these facilities to get help doing all these very complicated, very complex screening things. And of course also outside the house, the Conaway lab for their help with the STK19 experiment I showed you. And finally, I should say, we moved into this new glorious institute just a few weeks ago, and actually I'm in the process of trying to fill the lab. So if there's a very talented individual in the audience, I'm sure there are lots of them, please let me know. Thanks a lot for your attention.